Okay, excellent. Well, congratulations everyone for making it to the last talk of the day. I hope you can stay awake for the last 20 minutes. I hope I will sort of ignite some sort of great questions and passion of just touching on some of the new vaccine technology that we have in our midst. And I'm going to focus mostly on the pre-fusion F protein, the RSV vaccines. I'm going to touch a little bit about the mRNA vaccines and those that are in the pipeline. And then I'm just going to talk a little bit at the end about the new needle-free dermal high-density vaccine microarray patches, but really focus a bit more on RSV at the moment because it's just so topical. There's a bit of a delay. Okay, so I thought this paper was really interesting. It's a little bit old now. It's from the start of last year, but the R&D landscape for vaccines. I mean, obviously there are many more, over 1,000 candidates in development globally. But just looking at the top there, you can see most vaccine development happens in the US, followed by China. And then what's also interesting, if you look in the graph underneath, the B is there in the blue. Most of the vaccines, of course, are developed by industry, but in the yellow there you can see by the vaccine type, the academic institutions that do also develop vaccines. And this is sort of close to our heart at MCRI, thinking of the RV3BB rotavirus vaccine, and there are other fantastic stories of academia developing vaccines as well. But of course, the majority developed by industry. And then down the bottom, you can see there the types of vaccines in the pipeline, and the recombinant protein vaccines and the nucleic acid vaccines, including the mRNA vaccines, are really the two largest groups at the moment. So just touching on the mRNA vaccines, I know I'm speaking to the converted here, you know this, but I can't tell you how many times I explained how mRNA vaccines worked on a webinar through the COVID vaccine rollouts, because there was so much confusion and misunderstanding and fear that the RNA was going to be incorporated in the DNA and get into the nucleus and change people forever. And it's just really, simple actually, and it's exquisite technology, I think, how they actually work. But you can see there the virus at the top, the SARS-CoV-2 virus with the spike protein, which is encapsulated in a liquid nanoparticle, which then moves into the cell. Of course, the liquid nanoparticle dissolves, the mRNA is released and read by the ribosome, doesn't go anywhere near the nucleus and the DNA, and then that is how the spike protein is produced. The mRNA itself is also broken down and destroyed within days, as well as the liquid nanoparticles, so it can't be retained in the body for years or cause all of these, other conditions and concerns that people have about mRNA vaccines. And then, of course, the spike protein is released into the circulation to generate an immune response. And, this technology started well before the pandemic, more than 30 years previously. But, of course, the COVID vaccines were the first mRNA vaccines used in humans and developed very quickly. And this is what I found most extraordinary, was looking at the different infectious diseases that we now have mRNA vaccines in development for. And you can see them all listed there, and there are many more not captured in that pie chart. But also, incredibly, the other applications for mRNA vaccines for cancers, immunological diseases and other rare diseases. You can see there for the cancers, melanoma, brain cancer, non-small cell cancer, and so on. So quite extraordinary, the application for mRNA technology, and really such simple and clever technology. Now, in terms of the safety, we've all attended multiple talks through the COVID rollout about the different adverse events associated with the mRNA vaccines. I just thought I'd present this systematic review of 81 articles just to really show you the cardiac and vascular events observed with both the Pfizer and the Moderna vaccines, and the difference between them, I suppose. Of course, the risk profile differs by age group. We know, for example, with myocarditis, that occurs more commonly in young men. But you can see there for the Pfizer vaccine, thrombosis is by far and away the most common adverse event in the blue, followed by thrombocytopenia in orange, and then the stroke in grey, in comparison to Moderna, where you have stroke as the most common adverse event, followed by myocardial infarction there in orange, pulmonary embolism and myocarditis. But you could look at that and feel quite overawed and concerned about these vaccines when you actually look at the rates of these adverse events. They are still rare. You're talking about anywhere between 2 to 3 per 8 per million. And if you contrast that with the disease itself, this is where it's so crucial for us as clinicians and researchers, scientists, to really be able to convey the risk and the benefit to people when we're talking about adverse events. But certainly quite substantial cardiovascular events with these mRNA vaccines. And you can see from this review, there were many more deaths recorded with the Pfizer vaccine compared to Moderna. So I'm now going to move on to RSV, and you heard about the economic issues this morning and those things that we need to weigh up from an economic point of view. I'm really just going to touch very briefly on the burden of disease of RSV, which is obviously substantial in infants. We know that nearly 70% of infants have had RSV by the time they turn 1, and nearly all by the time they turn 2. Premature infants obviously have a much higher hospitalisation rate, up to 3 times higher than term infants. And we know that more than 75% of RSV disease hospitalisation occurs in full-term infants. The risk is much higher in Aboriginal babies. But also, and here just looking at those rates again by age group, you can see the graph on the left there, much higher rates of disease in infants 0 to 2 months in the navy blue, and then you can see the other age groups, but also in children 2 years to 5 years on the right, and then that huge spike of disease in the over 65s. And that's really important when you think about the program and the questions and what's facing us now in terms of what we're going to do. Really important to think about the societal cost of RSV. We know that RSV is the leading cause of hospitalisation in young children, and that estimate here, the societal cost of hospitalisation of Australian children under the age of 5 is estimated to be 193 million per year. So definitely a substantial burden of disease. And here just the comparison of RSV burden of disease to flu, obviously not as high as flu when you look there at the medical practitioner encounters, hospitalisation and deaths, but still substantial. So how do these new RSV vaccines work? Well, you can see here this beautiful photo of the RSV virion, and you can see the targets for the neutralising antibodies there. There's the attachment protein in green and the fusion protein in red, and both of those are the targets for the current RSV products. But this is really that understanding of the pre-fusion versus the post-fusion structure of the RSV virion. And you can see there that the pre-fusion, the red and the orange, have the highest neutralising potency. And it's that pre-fusion structure. The antigen has been engineered to retain that conformation, to display those potent neutralising epitopes in these current vaccines. And I think the question was asked earlier, why has there been such a long delay in developing these vaccines? And as Cantor said, it's because we haven't been able to actually have the antigen engineered to retain that pre-fusion shape and expose the red there in particular. So this is really the landscape that we're looking at the moment in terms of the RSV therapeutics. There are 2 vaccines currently available for adults over the age of 60. And then in infants, we have this really interesting program challenge of an RSV vaccine to be given to pregnant women, and also the long-acting monoclonal antibody that's now coming into the landscape to protect infants after birth in that first year of life, given the burden of disease that I just showed you. So Abruzzo is the bivalent RSV prefusion F vaccine, which doesn't have an adjuvant. It contains 60 micrograms of each of those prefusion proteins. So it's a 120 microgram dose. It's a single dose in a 2 ml vial. And at the moment, this is being considered at the moment in Australia for adults over the age of 60, but not yet approved. And in terms of the data to support this, you can see here really quite high vaccine efficacy against RSV-associated lower respiratory tract disease there, 88% in season 1 and 79% in season 2. And then on the right there, you can see some of the safety data. And really the thing to be aware of are these inflammatory neurological events. In this trial, there were 3 events detected in the vaccine group and none in the placebo group. Now, in comparison, the second RSV vaccine that is going to be available for adults, and this has been approved in Australia, is the efficacy vaccine. And you can see that's the single antigen, 120 micrograms, but this is an adjuvanted vaccine with the AS01 adjuvant system, similar to what was discussed this morning by Tony. And in terms of the efficacy data for efficacy, you can see very similar RSV-associated lower respiratory tract disease around 83%, but a little bit of a bigger drop down to 56% in season 2. And again, similar to the Abruzzo, you can see the 3 inflammatory neurological events that were detected in the vaccine group. So these were predominantly cases of Guillain-Barré syndrome, and all of the cases had an onset within the 42-day risk window post-vaccination. This was in the CDC surveillance, and the significance of sort of 1 to 2 cases in the safety database of 15 to 26,000 people is unclear. We know that the population rates of GBS increase with age, and very clearly we need post-marketing surveillance as these vaccines come into the market for older adults to really understand this and track this better. So in the US, both these vaccines are currently being used by adults and were recommended and approved mid last year. So in terms of the landscape, again, now I'm just going to turn to the monoclonal antibodies for infants. We know that there has been the availability of short-acting monthly monoclonal antibodies for high-risk infants, and now we have a fantastic new development with the long-acting monoclonal antibodies for infants in comparison to the option around maternal immunisation, which I'll talk about now. So this is the key trial that we need to be aware of, the Matisse trial, which was a phase 3 double-blind trial, just over 7,000 maternal participants across 18 countries. These were pregnant women between 24 to 38 weeks gestation, randomised to receive the Pfizer bivalent RSV prefusion F vaccine or placebo. And the highlight or the sort of headline finding was that it was effective against medically attended severe RSV-associated lower respiratory tract illness in infants. And I've summarised that data here. You can see there on the left, medically attended severe lower respiratory tract illness within 90 days of birth, VE of 82%, but it was not significant for medically attended RSV-associated lower respiratory tract illness with a VE of 57%. But the big question here is around safety with the maternal RSV vaccine. And in this particular trial, there was no signal around safety, particularly around prematurity. You can see there on the right, that is summarised. They looked at those issues around developmental delay, low birth weight, and prematurity. And just breaking that down a little bit further, you can see there were 201 babies who were born premature in the vaccine group, versus 169 babies in the placebo group. So that was only a difference of 32 infants. So it was not significant, but there still was a difference. And in the previous GSK maternal vaccine trial, there was a greater difference. And I think without going into too much detail about this finding, I think really it's going to be very important that we watch this issue around prematurity, and this has influenced the recommendations made around this vaccine in terms of who it should be given to. So you can see that it's been licensed by the FDA in Argentina, by the EMA. The FDA approved this as a single dose for pregnant women who were between 32 to 36 weeks gestation, which of course has quite clear programmatic implications. Only being able to give the vaccine to women who are 32 weeks. Other countries are looking at that differently, reducing that recommendation down to 28 weeks or lower. But I think in terms of what we know about confidence in vaccination and maternal vaccination program, we don't want it to impact the other maternal vaccines. If a prematurity signal does become more evident, then we just really want to reduce the risk of the impact of that vaccine safety signal. So you can see there the recommendations. The ACIP, as I said, have made that recommendation 32 to 36 weeks. In the UK, they've made a recommendation with no gestational age restrictions and an all-year administration program. And at the moment, the WHO SAGE group is really looking at how this will work in low and middle income countries and the recommendations to be made there. So then just briefly, I'm going to touch on nirsevimab, which is the long-acting monoclonal antibody, and the data supporting efficacy there. So you can see really nice impact of 77% on medically attended RSV-associated lower respiratory tract infection, which again is just really fantastic to have another therapeutic in the program. So the ACIP have approved, or the FDA, sorry, approved nirsevimab in July last year, and it's been recommended for all infants under 8 months born during or entering their first RSV season. And then for higher risk infants entering their second season, so from 8 months through 19 months, they may be able to access a second dose in their second year of life. And obviously really important that we think about and target the infants who are at higher risk. And regulatory approval has been given in the UK and Europe. And at the moment, as you can appreciate, there's a lot of careful thinking going on around how a maternal vaccine program would work in with this long-acting monoclonal antibody program here. And then lastly, in terms of the high-density vaccine microarray patches, this is really novel. This technology has been now in development and with trials happening over the last 10 years. These maps are categorised into 5 main types called solid, coated, dissolvable, hollow or hydrogel forming maps. And essentially they house an array of little microprojections and they breach the outer layer of the skin to deliver the antigen into the epidermal and dermal layers of the skin, and they have an applicator device as well. And they really are compatible with a huge variety of vaccine modalities. So the map that has got the most number of candidates in development are these coated maps, and you can see there the antigen coats the tip of the microneedle, and then the coated microneedles are applied to the skin and deposit the vaccine into the epidermis or down into the dermis, and then obviously you remove the microneedles and the vaccine remains in the skin. So these are one of the most popular choices for vaccine delivery. They have a very short wear time of just under, well, can be anywhere from 30 seconds to 2 minutes. Some of the other ones, the dissolvable versions, need to be left on for longer periods of time, 24 hours, 48 hours or longer. These have been evaluated for many different vaccine candidates, but mostly influenza. You can see the other ones listed there. And there are currently 3 phase 1 clinical trials for flu at the moment using these coated maps. And Vaxis is the company conducting a lot of this work based up in Queensland. And a lot of the data shows a significantly higher antibody response using the MAPS over the sort of traditional vaccine methods. So I think this is very much watch this space. And from what I can see in the reading that I've done, really only mild, moderate adverse events, no significant adverse events reported at this point. But you can imagine lots of programmatic delivery issues in terms of, in comparison to the usual sort of technology in terms of how this would be introduced into a program, used by children, acceptability and so on, but still a long way to go. So I'll stop there, and I just want to thank Kathy Newsall for some of the RSV slides that she sent to me for this talk. So thanks very much.